Are you a health enthusiast focused on longevity and optimizing your well-being? Do you carefully consider the supplements you take? Get ready, because new research suggests your daily dose of vitamin D might be doing more than just supporting bones and immunity. It could be directly impacting how your cells age. Let's dive into the fascinating world of cellular health. At the ends of each of your chromosomes are protective caps called telomeres. Think of them like the plastic tips on shoelaces. They prevent fraying and damage to your genetic material. As we age, these telomeres naturally get shorter with each cell division. This shortening is a key marker of biological aging. When telomeres become critically short, cells can no longer divide properly. This state, known as senescence or programmed cell death, apoptosis, contributes to the decline associated with aging. It's linked to an increased risk of various age-related diseases. Maintaining telomere length is therefore considered vital for healthy aging. A significant study, published in the American Journal of Clinical Nutrition, explored the potential influence of common supplements on this crucial process. Researchers specifically looked at how supplementation with omega-3 fatty acids and vitamin D affected telomere shortening over time. This research adds valuable data to our understanding of how lifestyle factors and nutrients might influence cellular longevity. For those of us focused on proactive health, these findings are particularly compelling. They suggest a potential protective role for vitamin D beyond its well-established benefits. The study utilized data from the large-scale VITAL trial. This trial involved a diverse group of adults across the United States. Participants were randomly assigned to receive daily supplements of vitamin D3, omega-3 fatty acids, both, or a placebo. The VITAL trial was a double-blind, placebo-controlled study. This rigorous design is the gold standard in research, meaning neither the participants nor the researchers knew who was receiving the actual supplements versus the inactive placebo. This minimizes bias and strengthens the reliability of the results. Participants were followed for approximately 5 years. Women in the study were at least 55 years old, and men were at least 50. This focus on an older adult population is relevant, as telomere shortening is more pronounced with age. To assess the impact of supplementation, researchers focused on telomere length in white blood cells, also known as leukocytes. These cells are easily accessible through blood samples and are commonly used as a proxy for overall cellular aging. Participants provided fasting blood samples at the beginning of the study, baseline, and again at follow-up visits. These visits included in-person assessments to gather other relevant health information. The researchers were able to analyze over 2,500 blood samples from more than 1,000 participants. Telomere length was measured at baseline, after 2 years, and after 4 years of supplementation or placebo. The results revealed a notable difference between the supplement groups. Participants receiving vitamin D supplementation showed significantly less telomere shortening over the study period compared to those taking a placebo. Specifically, in the vitamin D group, there was only minimal shortening observed at both the 2-year and 4-year marks. In stark contrast, the placebo group experienced substantial telomere shortening over the same timeframe. Interestingly, supplementation with omega-3 fatty acids did not appear to have a significant impact on telomere length in this study population. This highlights a potential unique benefit of vitamin D in this specific cellular process. The researchers also conducted exploratory analyses to see if the effects of vitamin D varied among different subgroups. These analyses, while considered hypothesis-generating, offered some intriguing insights. The positive effect of vitamin D on minimizing telomere shortening seemed particularly significant for participants who were not taking medication for high cholesterol. This suggests potential interactions between vitamin D and lipid metabolism pathways, or perhaps reflects underlying health differences in these groups. Furthermore, the benefits of vitamin D appeared statistically significant for non-white participants in the study. This finding is important and warrants further investigation to understand potential ethnic or genetic factors influencing vitamin D metabolism and its effects on aging markers. The study did not find a significant interaction with body mass index overall. However, participants who were not obese did show significantly minimized telomere shortening with vitamin D supplementation, suggesting that maintaining a healthy weight might enhance vitamin D's protective effects on telomeres. While these findings are exciting for health enthusiasts, it's crucial to understand the study's limitations. This analysis was conducted after the main vital trial was completed, a post hoc analysis, and the original study wasn't specifically designed to focus on telomere length. The study population was primarily white and over the age of 50. This means the results may not be directly generalizable to younger individuals or more diverse ethnic groups. Future research is needed to confirm these findings across different populations. Missing data, particularly at the 4-year mark, was another limitation. About 37% of cases were missing data after 4 years, which could reduce the statistical power to detect effects, especially for omega-3s. Unaccounted for factors could also have influenced the results. As with any study, these findings should be interpreted within the context of its design and limitations. The exploratory subgroup analyses, in particular, require caution and further confirmation. Experts not involved in the study have weighed in on its significance. Dr. David Cutler, a board-certified family medicine physician, noted that while the study is interesting, its clinical implications are currently limited. He points out the relatively small number of participants in the telomere analysis cohort and the focus solely on white blood cells. However, Dr. Joshua Quinones, a board-certified internist, expressed enthusiasm for the findings. He highlighted that while vitamin D is known for bone and immune health, this direct link to telomere preservation is particularly impressive. Dr. Quinones stated that the study provides a general idea that vitamin D3 could do more than just helping your bones, but actually slowing down the cellular age. He believes that if confirmed by future research, this could have significant implications for preventive medicine and potentially lead to new medical guidelines regarding vitamin D supplementation for healthy aging. The study authors suggest that the observed preservation of telomere length with vitamin D could potentially translate to a 3-year decrease in aging at the cellular level. This is a powerful concept for anyone interested in longevity. Future research is needed to explore the mechanisms behind this effect. It could involve vitamin D's influence on the enzyme telomerase, which helps rebuild telomeres, or its role in protecting against DNA damage that contributes to telomere shortening. However, it's equally important to consider the potential risks of vitamin D supplementation, especially for supplement users. While moderate doses, like the 2,000 units used in the VITAL trial, are generally safe for most people, excessive intake can be harmful. Vitamin D is fat-soluble and can accumulate in the body, potentially leading to toxicity. High levels can cause kidney damage and other adverse effects. Therefore, any potential benefits must always be weighed against known risks. This study offers exciting insights into the potential role of vitamin D in cellular aging, a topic of great interest to health enthusiasts and supplement users. It reinforces the idea that nutrients play a fundamental role in our long-term health. While more research is needed to fully understand the clinical implications and confirm these findings across diverse populations, it adds another layer to the known benefits of maintaining adequate vitamin D levels. Stay informed about the latest research and its implications for your health journey. Consider discussing your vitamin D levels and supplementation strategy with a qualified healthcare professional. They can help you determine if supplementation is appropriate for you, the correct dosage, and how it fits into your overall health plan, ensuring you balance potential benefits with safety. Your proactive approach to health is your greatest asset.